Right. This was folic acid that I've just told you, and the recap you've just heard. Now, another vitamin that goes along with it is called B12. That is vitamin B12. First of all, what is vitamin B12? So, this is vitamin B12. Just see the structure of B12. It has got 4 pyrrole rings. Structure somewhat resembles the structure of heme. But remember, it has got, it has got alpha methynyl bridges. Another one over here. Then one over here. But these two are combined together. So this is what we call as coring ring. Coring ring. Coring ring. So this is the structure of vitamin B12. What we're talking is vitamin B12 of the B complex series. It has got, this is vitamin B12. And so this coring ring is somewhat, it resembles the structure of heme, but the difference is that the 2 pyrroles, they are 2 of the 4 pyrroles are attached together, one, and second, it contains cobalt in the center, which is attached to 4 nitrogens of the pyrroles. You've got a pyrrole, so this has got attachment over here, so this is cobalt. In addition to this, it is attached to a moiety called as dimethyl benzimidazole group. So this is the total structure of B12. Now I told you in folic acid metabolism, B12 has got an importance that it relieves what? It relieves what? Folate trap. Folate trap. Folate trap. B12 relieves, बचाता है. It relieves folate trap. Because once the folic acid is activated to tetrahydrofolate, it is trapped with methyl group, HCH3 group, and this is called a folate trap. This is a folate trap, and because it is attached to methyl, so it cannot take place in 1 carbon metabolism. So 1 carbon metabolism will not take place, hence no formation of DNA. DNA is not formed. So B12 will come, B12 comes, this is vitamin B12, this will come and it will take the methyl away, forming methycobalamin CH3 B12, or called as methycobalamin. This is methylated B12. So this is methylated B12. So vitamin B12 relieves what we call as the folate trap. This is one importance of B12. That means if the folate deficiency leads to, say, 1, megaloblastic anemia, number 2, to anencephaly, number 3, spina bifida, directly, but deficiency of B12 by causing deficiency of active folate Yeah, by the inability of removing, this folate trap will definitely cause the above mentioned clinical problems. So B12 deficiency also causes megaloblastic anemia, indirectly megaloblastic anemia. It will also cause indirectly anencephaly, B12 deficiency, and it will also cause spina bifida defect. Plus, in addition, B12 causes derangement in mental abilities. And this derangement of mental activities plus the consequences of folate deficiency because of not relieving the folate trap is summed up as pernicious anemia. It's called as pernicious anemia. Now I'll talk about B12 in detail. The types of B12. Cyanocobalamin, methylcobalamin, deoxyadenosylcobalamin, and hydroxycobalamin. So these are the four important types. Number one, this one is the commercial preparation. This one is the commercial preparation, in which it is available in the injection form. Methylcobalamin is the most abundant form, and it is formed as a consequence of relieving folate trap. Deoxyadenosylcobalamin is present in mitochondria, and hydroxycobalamin is also present in traces. Methylcobalamin is the most abundant form. They were present in blood also, present in blood in combination with transcobalamin, transcobalamin 2. So these are the types of B12. One is cyanocobalamin. It's a commercial form, water soluble. It's water soluble, commercial form. And then you've got the most abundant form in blood, and it's carried along with transcobalamin 2. Then we've got deoxycobalamin is present in mitochondria. Then hydroxycobalamin is present in traces. Next, how is it absorbed? Well, B12 is taken in the diet, goes to the stomach, then it goes to the ileum, this is large intestine, So it is absorbed in the ileum. It is absorbed in the ileum. So if this was B12, this is cobalamin, B12, taken in the diet, cobalamin in the diet. It attaches to an intrinsic factor, a protein, intrinsic factor, that's a protein secreted by parietal cells. And then it forms a complex. This complex would come all the way down and will get absorption of B12. Cobalamin in the diet is called the extrinsic factor of Castle, and this intrinsic factor is called the intrinsic factor of Castle. So this is how B12 is absorbed from the ileum. B12. I will repeat again. Cobalamin is taken in the diet. It attaches with intrinsic factor secreted by from the parietal cells of the fundus. So it comes from here and then it is absorbed, forming a complex. There are some proteins called as R proteins, and then it goes to the ileum from where it is absorbed. Once it is absorbed, then it is methylated by relieving the folate trap, and so it causes further chemical actions. What are these? This was about the absorption. So remember anything? Note, if the intrinsic factor is destroyed by, for example, gastritis, by operative procedures of the fundus, because this is there in the fundus area. So, if there is gastritis or there is an operation, then the loss of intrinsic factor will lead to the absorption of B12 will be reduced. Also, if there is infection with a worm called as Diphyllobothrium latum, it will also cause deficiency of B12. So you can say deficiency of B12, or if there is some operative procedure of the ileum, over here there is some defect, again there will be deficiency of B12. So when there is deficiency of B12, then it will be unable to relieve the folate trap, and all the deficiency of folic acid will also manifest itself. So I am repeating again and again. And also, in addition, to fulfill the criteria for pernicious anemia itself, the low levels of B12 would cause defect in intellectual function. Thereby, megaloblastic anemia plus all the developmental defects of folate deficiency plus this mental ability, which is directly as a result of low levels of B12, will make the condition pernicious anemia. If there is only folic acid deficiency, there is megaloblastic anemia. But if there is B12 deficiency, there will be pernicious anemia. So if the B12 is deficient, it will lead to pernicious, P-E-R-N-I-C-I-O-U-S, pernicious anemia. So what is pernicious anemia? Pernicious anemia contains folic acid deficiency plus intellectual disability of the brain, mental ability. That is, pernicious anemia contains two things: folic acid deficiency, all those three things, plus mental intellectual disability. So this is called pernicious anemia. B12 deficiency causes pernicious anemia, while individual folic acid deficiency causes only megaloblastic anemia, plus developmental anomalies in the pregnant female. So this point should be clear again and again. So pernicious anemia is caused by the deficiency of B12. It's called pernicious anemia because the CNS involvement is also there along with anemia secondary to folate deficiency, etc., etc. Thereby, when you treat megaloblastic anemia, you give both of these things together. That is, folic acid is given, also B12, vitamin B12 is given. Folic acid to the tune of 300 to 400 micrograms per day. That comes to 0.3 to 0.4 milligram per day. 300 to 400 micrograms per day, okay? And B12, 3 to 4 microgram per day. That is 300 to 400 microgram per day is the dose of folic acid, as compared to 3 to 4 microgram for B12, so it is also given. Usually B12 deficiency does not occur. Why? Because the liver has abundant stores of B12 sufficient to last for 3 to 4 years. 3 to 4 years. But still, to avoid any confusion, to avoid any cover-up, whenever there is megaloblastic anemia, it is wiser to give folic acid plus B12. Folic acid in the dose of 300 to 400 microgram per day, and B12 in the dose of only 3 to 4 microgram per day. So remember, 300 to 400 microgram per day and 3 to 4, only 3 to 4 microgram per day for B12. Now, what is the biochemical action of B12? Remember, methylated B12 is the most abundant form in blood, I have told you, and it is carried along with transcobalamin 2. Transcobalamin 2, a protein, carrier protein. And it is also abundant in liver, where it is attached to transcobalamin 1. So in blood, it is transcobalamin 2. Now, what is the mechanism of action? What is the biochemical role for B12? Biochemical role. One, it relieves folate trap. Number two, it causes the conversion of homocysteine to methionine. In the liver. it causes the conversion of methyl malonyl CoA to succinyl CoA. In fact, so it relieves folate trap, but it has got only two biochemical reactions. In the liver, if this is a liver cell, then you've got mitochondria. So over here it becomes methylcobalamin and it converts homocysteine into methionine. Then it would enter the mitochondria. This is mitochondria. This is mitochondria. And in the mitochondria it converts to deoxyadenosyl cobalamin. And deoxyadenosyl causes the conversion of methyl malonyl CoA to succinyl CoA. So, one, it helps in the formation of methionine. Second, it helps in the formation of succinyl CoA. Now remember, if there is deficiency of B12, so no folate trap, no methionine, and this reaction would also be blocked. So there will be three blocks. A, folate trap, that means leading to subsequent deficiency of folic acid, leading to megaloblastic anemia and developmental defects, told you. Second, this will be blocked over here. There is a defect. So no methionine, no succinyl CoA. But there will be excess of homocysteine, leading to homocystinuria and also methyl malonic aciduria. So these are the three things that would happen if there is B12 deficiency. And the treatment I've told you is that you give both of these drugs together, and so that all the things are covered. Folate deficiency is covered and also pernicious anemia is covered. One thing is that the brain changes can may be irreversible, but still in the early stages mega doses would do something better. So that was for cyanocobalamin. Now talking about because we are talking about water-soluble vitamins, I will talk about thiamine, because this was important just to emphasize the role of folic acid in the development of fetus and B12, so I considered these one. Started with these ones. Now coming back to vitamin B1. Vitamin B1 is called as thiamine. Thiamine. Well, The structure of thymine got someone like structure of a pyrimidine. And it's attached to a thiazole group. Then it's attached to, this is thymine. Then it's attached to one phosphate and the other phosphate. When these two phosphates are there, then you get the formation of thiamine pyrophosphate. This is the active form. This is thymine pyrophosphate. So thymine is vitamin B1. Vitamin B1. And where is it located? If you take this one as grain, so it is the undersurface of the grain, undersurface of the covering, undersurface of the covering, which contains B1. So this is the grain. So it is the undersurface of the covering. That is brown flour. Brown flour contains B1, यानी चक्की का आटा, चक्की का आटा, and not white flour. White flour does not contain, because उसमें से husk निकाल दिया जाता है, husk grain kernel का, under surface of the covering, husk. There's a brown bread and brown flour, brown. This is contains B1, and B1 is very important. Now what is the importance of B1? So I tell you, B1 is present on the undersurface of the covering of the kernel of the grain, of any grain. Be it wheat grain, barley, maize, oat, इन सब के जो छिलका है उसकी अंदर की तरफ B1 होता है. तो अगर आप उसका छिलका उतार देते हैं और आप व्हाइट फ्लोर निकाल लेते हैं या चावल का हस्क उतार लेते हैं, पॉलिश राइस करते हैं, तो उसका जो कवरिंग तो उतर गई, उसके ऊपर जो ब्राउन अंडर सरफेस थी वो भी उतर गई, तो B1 सारी उतर गई. एंड व्हाट यू आर ईटिंग इज a diet without B1. So B1 is present in the under surface of the covering of the grain. This is important. Now I tell you one thing. Why is it important? Because thiamine pyrophosphate takes part in essential metabolism. I come back again. How? Because we know that all cells need glucose. Glucose is converted to pyruvate. This is called as glycolysis. This is glycolysis. Then this pyruvate enters mitochondria. This is mitochondria. enters mitochondria. Pyruvate comes inside, yes. There it is converted to acetyl CoA. And it's also converted to oxaloacetate. Then you've got certain reactions that converted to citrate, is converted to isocitrate, cis-aconitate, oxalosuccinate, and repeat again. Pyruvate comes inside, is converted to oxaloacetate and acetyl CoA. Then it gets the formation of citrate, then cis-aconitate, isocitrate, oxalosuccinate, then it forms alpha-ketoglutarate. Alpha-ketoglutarate. Alpha-ketoglutarate, yeah. It is this one, alpha-ketoglutarate. Then succinyl CoA, then you've got succinic acid, then you've got fumaric acid, then you've got malate, and then you've got oxaloacetate. This is from